great pleasure to be here today to talk to you at this very interesting meeting in this very engaging and interesting city. And it's a particular pleasure to be talking about bacteriophage therapy due to the fact that the overall theme of this meeting is on solutions for the next decade. And the chief medical advisor to the UK recently said that one of the major problems we have facing humanity is resistance to antibiotics. She said it's a problem as big as terrorism or global warming. We have many bacteria now that we're one antibiotic ahead of, or two, or some that are resistant to all antibiotics that are known. So where are we going to find our next generation of antimicrobials? And part of the solution could lie in bacteriophage therapy, and that's what I'm going to talk to you about today. I'm going to give you a brief introduction to the subject, followed by an example of how you would set up a program of bacteriophage therapy for an organism you're interested in. And then our other three speakers are going to tell you about specific aspects of phage therapy that their work is concerned with. So just to tell you, first of all, what a bacteriophage is, it's a virus that specifically infects a bacteria. So can you see this bacterial cell here? It's decorated by little bacteriophage particles. And one of these bacteriophages has already attached to the cell, injected its DNA, and that bacteria is now no longer a bacteria, really. It's a little bag of viruses, where the viruses are waiting to pop out and kill more bacteria. So all bacteria have these natural viruses that infect them, and they're generally specific to a species. So in the same way, if you catch a flu virus, your dog won't catch it. Viruses that infect bacteria are very specific to the different bacterial types. And there's been an increased interest in this area, not just from doctors and healthcare practitioners, but also from the general public who are trying to cover this area of where we're going to get our next generation of antimicrobials from. So this was just a picture that was taken by a journalist recently in the UK who was covering some of the bacteriophage therapy work in my lab. So, what is the problem? Well, here we have a graph showing antibiotic resistance. This was just one study that was done over a 12-year period. And can you see these bars? These bars represent 3 different bacterial species, showing the amount of bacteria, the proportion of bacteria that this particular doctor found in a study that were resistant to antibiotics. And you can see the proportion is going up and up. And this is due to our overuse and our misuse of antibiotics. So we're exposing bacteria to antibiotics in a not particularly sensible way. We're really encouraging resistance. And at the same time, this is the amount of new antimicrobials that are coming online. You can see the proportion is really going down. And the reason for this is that to develop new antimicrobials is not as cost-effective as it is to develop medicines for chronic conditions. So if you have a bacterial infection, you have it for perhaps a week, 2 weeks. So there is less and less investment in new antibiotics. So where are we going to find this next generation of antimicrobials to treat infection? Well, part of the solution might lie from exploring bacteriophages, so these natural enemies of bacteria. And they could either be used by exploiting whole bacteriophages, so finding bacteriophages that target a pathogen that you're interested in, or you could work out how a bacteriophage kills bacteria and use those proteins that the bacteria makes in order to do the killing. So a bacteriophage has to get into a bacterial cell. So if you can figure out how it's doing that, you could use that property rather than using a whole phage particle. So they're the 2 main approaches that can be used. The idea of using bacteriophage to treat bacteria is not new. In fact, it completely predates antibiotics. Bacteriophages were discovered 99 years ago, and they were used until antibiotics were discovered. They were used in a number of studies and to treat a whole range of bacterial diseases, mainly by this chap up here, Felix Durrell. And interestingly enough, until the 1970s in France, you could still buy several hundred different bacteriophage preparations to treat bacteria. So although it wasn't a mainstream option, it was still used in some places. The place that's probably the most famous for bacteriophage therapy, if any of you have heard of it, you possibly associate it with the Republic of Georgia. And this is because there was a young Georgian scientist, went and trained with Felix Durrell, learned about bacteriophages, and set up this center here, this very famous Eliava Bacteriophage Research Center, where bacteriophages were manufactured and used a lot. This was the peak of this production was in the 1980s when 1,200 workers were producing 2 tons of bacteriophage preparations every year. And they're still used routinely to this day in clinical practice for a whole range of different conditions. So, where do you get phages from? Well, they're absolutely everywhere. They are the most abundant organism in the biosphere. So, for every bacteria that exists on Earth, all 10 to the 30 of them, there will be between 10 and 100 bacteriophages which can infect that bacterial cell. And this picture here, I think, illustrates that fact really nicely. This is just a teaspoonful of seawater that's been stained with a stain that stains DNA. And you can see all these large particles are bacteria, and these little ones here are bacteriophages. To have a wee zoom in and see what they look like this. These are the 3 main families of bacteriophages. You can see they have slightly different tails, as these have a contractile tail, a long siphon tail. These just have very, very small tails. And most viruses that infect bacteria conform to one of these 3 morphologies. They have 2 different modes of infecting bacteria. One is this mode here, where they infect, take over the cell, and viruses are released. And this can take anything from maybe 20 minutes to several hours. And in nature, the implications of this life cycle, I've written on the side here, they're shaping the population dynamics. So all bacteria have these viruses, but often they're just specific to a subset of strains within a species. So by killing that subset, other strains will then evolve, will then become abundant, and viruses will then evolve to attack them, and so on. So viruses and bacteria are involved in this continual arms race. And essentially, they're tweaking, shaping the population genetics. They're also important for the long-term evolution of bacteria, because in this stage here, what they can do is they can stuff their heads full of DNA, not from the viruses, but from the hosts, and they can shunt that into another bacteria in a non-harmful way. So they're important for long-term evolution. And as they kill bacteria, they release nutrients, which is important for the cycling of nutrients. Now, this is a subset of viruses that we're interested in using therapeutically when we're talking about using whole viral particles. There are another side to bacteriophages, which is shown here. And what they can do is they can also inject into the bacterial DNA, and they can integrate their DNA into that of the bacteria, and they can remain stable there for many thousands of generations. But as I say, the rest of my talk and the talks that will be given in this session today are concerning viruses which are capable of killing bacteria. Now, when might they be useful? Well, I'm going to give you 3 examples of that. The first is if bacteria are resistant to all antibiotics that are known, then they're bound to be useful, because what can we do? So if we have a bacteria that's shown to be resistant to all antibiotics, bacteriophages could offer another alternative. Another way that they're useful is in areas that are hard to reach. So for example, in a biofilm, you have a lot of mucopolysaccharides secreted by bacteria, and antibiotics actually find it quite hard to penetrate into that environment. Bacteriophages have enzymes that can degrade these biofilms, so they can be very useful then in that context as well. And the third example of where they're very useful is because they're so specific. If somebody has an infection that whereby antibiotics might complicate it, for example, an intestinal infection, the antibiotics that you take will probably make you feel quite groggy, and as they knock back all of the other bacteria which we actually need. A virus is so specific that it would just take out this one bacteria that's causing the infection. So these are the three examples of where bacteriophages could provide very useful treatment options. So if they're if they're as good as this, why are they not being used already? Well, the main reason is, after antibiotics were discovered in the 1940s, bacteriophages were sort of sidelined. They were seen as not really necessary. They're very difficult to use in that they're so specific. It's difficult to, you really have to know your enemy. You can't just give a patient an antibiotic. So their specificity was seen as a disadvantage. And antibiotics was seen as a sort of, as a cure. Therefore, it was felt not, in the West certainly, the use of bacteriophages to be developed as a therapeutic was really, research in this area was practically really diminished. Also, there were problems at the time in the 30s and 40s of overselling bacteriophage preparations. So they promised the world, they promised to cure all sorts of even viral infections, which they couldn't possibly do. There's also problems with standardization of a product. So biological is harder to monitor compared to a regular drug. And there were issues with toxins in the preparations as well. And the final reason, I think, was probably these political associations with Eastern Europe. Phage therapy was seen as a thing that these crazy Eastern Europeans tried. It didn't work. So when I started working in this area a decade or so ago, some of my colleagues were very much of this ilk. But I've really noticed a shift in change of acceptance as we have this growing need to study them further. So what can we do in this day and age? Well, we have a lot more tools than were available 100 years ago. So we can sequence genomes of both the host and the phage. And that can really help us in understanding the dynamics of the interactions, and it can help us with making sure we have a pure product that's not going to introduce toxins into a patient. And also it can help with standardizing a product. And Marta Vives is going to talk to you more about this subject in her presentation. We know an awful lot more about the bacteria themselves now. And this can really help us in many ways, both from a therapeutic point of view and a diagnostic point of view. Now we know our targets, we can explicitly choose phages which can hit the right bacterial targets. And Ayman El-Shabini is going to talk to you more about the diagnostic aspects of bacteriophages in his presentation. And the other thing that we have now that's much better than it was 100 years ago is we have a lot of advances in technology in terms of the production of phages and in terms of improving their delivery. So our final presentation of today is going to be from Hani Anami, who's going to talk to you about some really innovative ways of delivering phages to the right place. So what I'm going to do next for my presentation is give you an example of how you go about the steps that you need to take if you want to use bacteriophages to target a bacterial pathogen. And I'm going to just tell you about one of the projects that I've really focused on over the last 6 or 7 years, and that's of Clostridium difficile. And if you look really, really closely, there's a Clostridium difficile cell. You can see a little bacteriophage just hooking onto it there underneath all these flagellae. And Clostridium difficile is an interesting target in that it's the main cause of infectious diarrhea in the Western world. It destroys the colon, and it causes death in about 10% of all the patients it infects. And the numbers are really quite huge. And last year in the UK, there were 24,000 cases. The economic burden in the US is estimated to be $10 billion a year. In the EU, it's estimated to be 8 billion euros. And we've got, on top of Clostridium difficile, to a certain extent, and that rates are falling, have been falling slightly over the last 6 years, and that's through increased measures of hand hygiene and a lot of decontaminating of wards and awareness of the organism. So it causes a real problem, actually, because it makes these spores. So when it becomes unhappy, it just makes spores, and these spores settle so slowly. It takes 3 hours to settle a meter. So once a hospital has them, it's very difficult to get rid of it. And there are still endless strains of C. difficile emerging. There's over 350 recognized strains, and there's 2 antibiotics that are routinely used to treat it, and 1 new one. So there's a need to develop new antimicrobials, because this is indeed one of the diseases where antibiotics are associated, the use of antibiotics initially are associated with the actual clinical symptoms of Clostridium difficile. Okay, so it's a big problem. How would you use phages? Well, you could use them just instead of antibiotics, or you could use them prophylactically. There are a subset of C. difficile patients that relapse endlessly, so these patients could be given a little cocktail of bacteriophages, either in a capsule form or the form of a drink, and the phages would migrate through the gut into the colon, where they would then target the Clostridium infection. And you could potentially use them in combination with other therapies as well. Okay, so what do you have to do to do this? Well, first of all, you have to find your phages, and then you've got to determine that they kill what you want them to kill. You can characterize them, make sure their genomes don't contain anything nasty, study the dynamics, and then check that they work not just in culture, but in model systems. So I'm just going to walk you through these phages, these stages even. So if you want to use bacteriophages, you obviously have to know something about the diversity within the strains that you're trying to target and kill. So this is what I've been building up a big collection of Clostridium difficile. So this is my sampling ground. This is my local hospital, the Leicester Royal Infirmary, and I've been gathering patient stool samples from everybody who gets this disease. We still have about maybe 20 a week of people coming in through Leicester with Clostridium difficile. So I isolate the strains, and here we can see this is strains on a plate. They under an ultraviolet light, they glow this nice yellowy green color. I've also been isolating strains from the environment as well. So I've amassed a fairly large strain collection of which I've sequenced a proportion. And then this was the bit that I thought would be much easier than it was, actually. This is the section when so I thought it would be quite easy to find Clostridium difficile phages, and I chose to work on this because I could see there was very little work done in this area. There's several research groups working on MRSA, Pseudomonas, and other major pathogens in the UK, but there was much less work done on Clostridium. And I thought it was just people hadn't really looked for phages properly. But then 4 years later, after a very dedicated team, we managed to find bacteriophages. And can you see these little holes in the plate? This is where a bacteriophage has killed some bacteria, and then the bacteriophages are spreading through that plate. So these are my students and this is my postdoc and PhD student, and who've been very instrumental in this work. Now, these are my top 26 bacteriophages. So after this period, we have a lot of viruses that infect this organism, and you can see they share two of the main morphologies I showed you about, the long contractile tail and the large siphon-like tail. And the first thing that we did was actually look to see if the phages can infect the right strains of Clostridium. In other words, the strains that are circulating. So all I want you to see here, this is just the colors represent the different strain types, and these are the strain types that were circulating over a 3-month period in my local hospital. So you can see there's lots of this strain and a few of many other strains. So this was my student Anisha, who then took my 26 phages and checked that they could infect these strains of clinical relevance, and luckily they could. Here, this shows a host range analysis. Probably easiest actually if you look on this plot. This is phages against strains, lots and lots of strains. And you can see that some phages are really, really narrow, like this phage here this phage here can only infect one subtype of one strain, whereas this phage here can infect many strains. So for therapeutic purposes, it's useful to take phages that can infect many strains within the species of interest. And here we have, it's quite a bit dark in this. But if you look closely, you can see this is a lot of different bacteriophages attaching to a Clostridium cell. And the first thing that we do is just looking to see how well they kill in a broth culture. So here's an example of this. I mean, you can just see this is a control. This is a Clostridium growing without bacteriophages added to it. This is the log of the number of colony-forming units per mil. And you can see that this phage, after 4 hours, has killed effectively all the bacteria, whereas these phages here are less good. They've had a little bit of an impact, but there's still a lot of bacteria left. So we start off just by seeing, and you can see these are nicely cleared broth cultures. And then you'll be using bacteriophages in a setting where the bacteria aren't just growing optimally in broth, so it's important to make sure that they can work in other settings as well. So here we have a graph showing that they work in an artificial gut model. This is where you spike a stool culture with Clostridium difficile and then add phages. And it just shows the phages can replicate in complex systems. So that was one thing that we did. I've also been developing a system to study Clostridium bacteria and phages interacting on epithelial cells, because that's where we'll be needing to use them clinically on the gut epithelia. And you can see in this diagram here, this is where cells this way Clostridium is growing on epithelial cells, and in here we can see when we've added the bacteriophages to the epithelial cells, we have a several log difference in terms of the amount of Clostridium that's present. So that's good news. The bacteriophages can also clear in those conditions as well. And this just shows that the phages are actually replicating locally. And the final plot here shows that, which is quite important too, shows that this is the effect, the cytotoxicity of Clostridium using lactose dehydrogenase as a marker. So basically, you look at this massive peak here, this shows that the cells are very unhappy because of the toxins that Clostridium is releasing. And when you lyse those cells with phages, there is no there's a much lower toxicity. The toxins aren't causing a problem to the human cells per se. I've also moved into animal models. And here we can see an example of animal work where this is in a very tight model. With Clostridium, you have to use hamsters because mice are not susceptible to the toxins of Clostridium. And in this model, my Glasgow collaborator knows if she gives 5,000 spores of Clostridium to a hamster, they die at this time point. Now, when we've added bacteriophages, we've doubled the life expectancy of the hamster. So I was a little bit disappointed when I saw these results, but my collaborator assures me that when you see such a big difference in an animal model, it's an intervention that's really worth taking forward. So we've been doing that. We've done a lot more of this work now with using different phages and different strains. And I should say, actually, that this work is being funded by a commercial company who had seen me present some of this work at a conference, and we started to talk about how we could take this forward. And I've been working with them because they have bacteriophage therapy programs for other bacteria, and they're really pushing forward a regulatory platform. And my Clostridium work fits nicely into their program. So it's a nice example of academic and industrial relationship. And this company is called Amplify. Now, hopefully, therefore, I have taken you through the various steps that you need to do should you want to look at bacteriophages to target a pathogen. So you can see I've gone some way to establishing that the phages have the right host range. I've studied their genomes. I haven't had time to go into the genomes in much detail. I've talked about just studying different models that have been developed to study their interactions. And hopefully, if the animal work goes well, the company who's supporting this work are wanting to move forward into human trials. And then we've also been optimizing the production of these bacteriophages as well. That's also important. So in conclusion, hopefully I've convinced you it's an exciting time to be a phage biologist and to be looking at bacteriophages as a necessary source of new antimicrobials. There's still a lot of basic science to be done. So we need phage banks for particular pathogens that are a problem. We need to determine the biology, the interactions that underpin the development. Obviously, we need clinical trials done under conditions that are accepted everywhere. And this clearly needs collaboration between academia and business. And there's a regulatory pathway is needed for the therapeutic usage. Now, the regulatory framework is getting better. For example, in the food industry, the FDA have recently regulated phages for the use of listeria in hot dogs. So if you buy hot dogs in the United States, they may say on them contain phage preparations. So that's phages that target listeria. And they've also approved, phages have also been approved for phage food usage in cheese in the EU. So the regulatory framework in the food industry is ahead of that in the therapeutic usage. However, the engagement that I've had with the regulatory authorities is that they've been extremely helpful. And my last point, which is hopefully of relevance to many of you young students in the audience, is you can see that this subject really needs engagement both with microbiologists, phage biologists, and doctors, pharmacists, engineers. It's not going to, the development of phages needs input and collaboration with many disciplines. So I'd just like to acknowledge my laboratory. This is why, one of the reasons I'm very happy to be in Alexandria. This is typical conditions in the UK at the moment. Look how warm my students look. And various collaborators. And I think this talk is going to be available on the webcast. So if any of you want to hear some information about the work in Arabic, you can click on this web link, because the BBC Arabic division were also interested in this work and came and made a really nice interview. So you can watch it in Arabic there. So thank you very much for your attention, and I'll hand over to Martha. So now I'd like to introduce my next speaker, who's Marta Vives, and she has come from the University of the Andes, and she's going to tell you about some of the. Oh, should we have? Yeah, should we do that? Oh, shall I take? Yeah. Marta's corrected me. I think, is there time for, is there one or two quick questions before we move on to Marta's presentation? We'll hopefully have some time at the end for, yeah.
At the end?
There's one question here, I think, this person. Somebody got a microphone.
Marta?
Assalamu alaikum. I would like to answer about targeting of the phage to the host cell. I know that it's a kind of viral DNA. How we could be sure that it would go in its right forward straight? It wouldn't attack other non-pathogen cells.
Yeah, it's a very good question, and it's often the first question that people ask. How do we know that these viruses aren't going to attack us? And the outside of the human cell is so different from the outside of a bacteriophage cell that there's no danger of the bacteriophage being able to put its DNA into a human cell. They're so specific even within bacterial species. So something that infects E. coli won't infect a streptococcus. So there's no danger of them being able to attach to the surface of a human cell, which it would need to do before it could inject its DNA. So we should be safe from that point of view. Yep.
Thank you, Marta, for the presentation. I find myself ought to ask a question about the Clostridium Difficile since it's spore former. I wonder if you, by any chance, came across what is the interaction happening between the phage and the spores, because myself, I work with the Bacillus series, as you know, and like it's kind of very much the factor that these kind of bugs, they survive in the environment for prolonged times is the fact that they form the spores. And I wonder if you came across how your phage are active against spores. Or
yeah, that's another nice question. We have tried, but they don't seem to have any direct, they don't infect the spores. I mean, yeah.
It's infecting through, like, when they are sporulated,
yeah.
the Phage DNA was being trapped before. But I personally looked in the literature about the Clostridium, and it seems that nobody...
Yeah. So Tarek was asking me, is there an interaction between phages and spores in Clostridium the way there is in Bacillus? We have actually just been doing these experiments, and they do get trapped in the same way, yeah. And when you can conceptually imagine in the next generation of phages, if you could modify them, making a phage that would potentially interact with the spores in some way or increase germination. But with spore formers, it's more of an issue. Anyway, I think in the interest of time, we should move on. We have more time for questions at the end. And over to Marta Vives.
Thank you, Marta. Salam alaikum. I just learned these words and I love them. Good afternoon to all. First of all, I want to thank the organizing committee for this invitation. It's an honor to be part of this incredible conference in this wonderful location, which has a special place in our imagination and dreams. This talk is going to be around the selection that we have to do when talking about the phages that we want to use in phage therapy. So that's the reason of the title of this talk, Not All Phages Are Equal. So, you already heard about the advantage that phages have. They are, they can deliver a lot of good things for the human society. They are natural controllers of bacteria that can cause problems. They are self-multiplying and self-limiting as well, because when the bacteria that they can attack goes down, they are obligate parasites, they also goes down. So they are specific, they are easily isolated, easily handled, no side effects, compatible with other therapies. I mean, the advantages are pretty obvious. However, I think that precautions must be taken, and we have to take that seriously. As Marta already said, there are many steps that you have to comply when you want to use the phages as a therapy. So we have some challenges, and we have to be very serious about these challenges. One of those is assure efficacy. What you want is that phage therapy works and lasts. Second, you have to reduce the risks associated to recombination, and I'm going to go back to that point afterwards. Also, Marta said about the quality control that we need to have about phage preparations. We have to validate in animal and human trials. And this is a very important step. We have to build and enforce regulations that allow the phages to be used. And also we have to work with public perception, because it's a virus. Yeah? So it's difficult to tell people, I'm going to treat you with a virus. So we need to work on that. My talk will be around these two, how is it? I cannot make it work. Okay, doesn't matter. So these two steps, because these are the first one, and all what goes after that depends on what we do in the first steps. So we have to establish very rigorous selection of the phages that we want to use. So, thank you. This one? Okay, good. So, in the first part, so if we want to assure efficacy, we have to work with lytic phages. And that was what Marta already showed regarding viruses that has the ability to kill bacteria. And this is what you see. You see a clear space when you don't have bacterial growth. And that's what a lytic virus does. It kills the bacteria, so you can even take this part, this is what we call a plaque or a plaque, and there you have an enrichment of the viruses that you want to select for. So in the beginning, you start with that, and that's very easy to start with that. So this is, again, as Marta said, an invitation for young scientists to work in phage therapy, because start with phage work is really easy. This is an agar plate where you can grow the bacteria, and then you go and isolate phages, and they are everywhere. See, almost every bacteria you can find phages against that. So you see this plaque, and you isolate phages that are lytic. You have to be careful, for example, with this other type of plaques that you can have, because probably this is a lysogenic phage. And we will talk about that a little bit later. You can see the difference between this plague, which is very clear, and this other one is kind of turbid. Yeah. After that, you go for a broad spectrum, a broad host range, because you already know that phages are very specific, so you don't want to have a phage that attacks only one bacteria, because in the environment or a patient or an animal, you don't know what species, what strain you are going to encounter. So you want to try to isolate phages that are able to kill several strains in the same species. And as you already showed from Marta's data, then you go for the infection curve. And this is exactly what you see here. I hope you can see it. This is the red line represents the growth of the bacteria after we add the phage. So if you remember, a bacterial growth curve should see like this, but when you add the phage, you see the growth of the bacteria diminishing, and these green and blue lines represent the phages. So it works. It really works. You can do it in the lab, and you will see it works. Another thing that you consider in these first steps is the virulence of the phage that you isolate. You probably want to work with phages that are needed in very low quantities but can kill a lot of bacteria, and that can be measured with criteria like low multiplicity of infection, a short latent period, high burst size, which means how many phages can a bacterial cell release. So, I already told you that phages work, and I love phages, but you have to be very skeptical. And what we did, being skeptical, to try to prove that phages work, was that, okay, this is the regular infection curve that you have for a phage and a bacteria. And they normally goes to 5 or 6 hours of incubation. We leave this until 8 hours. Yeah, so it really works. But I convince my students to work in this way. So this is working, I believe you, is great. But what happens if we just continue incubating for a longer time, bacteria and phages? And what we found was not surprising, but was the results that the students finishing the thesis never want to see. Was that the bacteria start growing again. And was the same for this another strain, and the same for the other strain. But that's absolutely natural, because bacteria and phages coexist. So there must be a way that bacteria circumvent the lytic cycle or the infection process or almost anything. Bacteria will prevail. So what we learn from this is that we must include this other criterion in the first evaluations that we do for the phages when we are trying to select phages for phage therapy. And this is the evolution of bacterial resistance. And this is not a trivial matter. Bacteria have a lot of mechanisms that allows them to be resistant to the phages. And here mentioned in very rough groups, they can, for example, prevent phage absorption or prevent DNA entry into the cell or degrade the DNA when it's into the cell, or start with another different mechanisms that are called abortive infection, which are one of the most wonderful things I've seen in my life, because it's kind of a social thing among bacteria. when a system for abortion infection starts, means that the bacteria prefers to die. And when it dies, the phage also dies. It's really interesting. So, but this is not what is shown here. I don't know if you can see it. It's another one. I'm in love with these bacteria and phages and everything because this is another wonderful system. It's called the CRISPRs. Maybe some of you have heard about that. And the CRISPRs is a system that allows the bacteria to have memory, just like an immunology. Bacteria can pick up some small pieces of the DNA of the phages and recombine it with its own genome, and when the bacteria again encounters some phage that's similar to that, it recognizes it and degrades the DNA. It's just amazing. So that's good for the bacteria, but that's not what we want when we are doing phage therapy. So one of the approaches that we have taken tried to understand how evolution of bacterial resistance to phages occurs and how can we circumvent this problem is phage dose optimization. That's, you know, for any drug you have to optimize the dose. And also for bacteriophage we have to do it because there are some cases well documented that shows that, for example, T7, which is one of the phages that is studied as a model and for many other reasons, some studies show that when you use more phages than needed, the bacteria enters a lysogenic cycle. If you go lower, you lower the dose of the phages, the bacteria enters the lytic cycle, which is the one we want. So it's an important issue. So we did that, and again, Marta made a wonderful introduction and just went every step I want to stress here, and this is another one. This is a work I like very much to present because it was performed with people from the chemical engineering department in my university. So this is biotechnology, and we need to collaborate with other persons because no one has the full background, academic background to address all the topics that we need to address. So what we did, we started doing some infection curves and growth curves and gave all this data to the group of Professor Andres Gonzalez, my colleague from Chemical Engineering Department, and he started to build models, mathematical models of the interaction, and then went for error minimization, selection of the best model for each phage bacteria pair, then do some other optimization with evolutionary programming, and we got like an optimal phage dose. Of course, they continue doing some other experiments with computers, and we got some in silico information that back up this optimal dose. Of course, when we have this dose, we went back to the lab, and the students in the lab tried to do, no, they performed, they did it very well. Some students with planktonic cells, and afterwards, what we obtained was that our phage dose was exactly the optimal. So this is an example of a lower dose. When we use a lower dose, the bacteria continue growing. When we use the optimal dose, the bacteria were killed. And we went one dose above. Also, we have problems, as I already told you, with T7. The next step with that was testing in mice. And this phage we were working with was against Pseudomonas aeruginosa. Pseudomonas aeruginosa is a well-recognized pathogen, human pathogen. And we tested in mice. This line you see here corresponds to the control group that was inoculated only con Pseudomonas aeruginosa. And these others here are those that were inoculated with Pseudomonas aeruginosa and the phage at different time points, 45 minutes after the bacteria, or 24 hours, or 48 hours. And we were able to recover almost all of the mice that were co-inoculated with the phage.
1 minute.
1 minute? Okay. The second part is design of the phage cocktails. As you heard, the bacteria develop resistance, and this comes from a very seminal paper studying the coevolution of resistance and between phages and bacteria. And we did that for some other model, Vibrio, which is a bacteria that kills shrimps. And we showed that when we use individual phages, we got resistance all the time with the 3 phages. But when we combine the 3 phages in one cocktail, the bacteria never obtain that level of resistance. So definitely, we have to use cocktails, phage cocktails. But we have to work on the selection of the phages and the proper assembly of the cocktails. Well, we also have to reduce, I'm going to be a little bit faster because time is running. We have to avoid this. That's the main reason we don't want to use lysogenic phages, because they can bring toxin genes to the bacteria if they recombine with the genome. So you remember this picture? We don't want to work with that, but that's very subjective. So that's the reason we decide to go for genomics. And we select one of the phages against Pseudomonas aeruginosa. I told you that saved the mice, because we were sure that that was a very good candidate for phage therapy. But in the end, what we got was information in the genome that shows that this is probably a lysogen, a phage that can do the lysogen cycle. Why? Because we found this C1-like repressor and CRO repressor. Not even like the virulence things, but this information may lead to the conclusion that this is a phage that is not suitable for phage therapy. So, genomics is important, but also we need classic work on gene function, because almost 75% of the open reading frames resulted to be hypothetical proteins. And this is all what you already know that we have to do. We have been working with animals models, as I told you, with shrimps. The mice study in the lab, but this is in a productive farm, and also with chickens, with poultry, because we thought that maybe we can change more rapidly the public perception if we show good results with animals. So, the important part of my talk is we need to use genomics in a very early step, because it saves time and it saves resources. This is a pipeline that a group of researchers, Meravishvili and collaborators designed, and this is a great effort, but you see here is the genomic work. So you went through all these to finally maybe realize that you cannot use that phage. So that's why we propose that genomics, which is now easier, cheaper, faster, can be in the very first steps. And the second one is that we have to work with cocktails, and we have to select very carefully the phages that we are going to use in the cocktails, mainly thinking about the evolution of bacterial resistance. So my conclusions are, phage therapy must be a science and technology-based development. And that's why I repeat that. It requires very well-trained scientists, because you need to know what you are working with. It has enormous potential. It has many advantages, but you have to be careful when are designing it, and it must to be followed up. So this is my group. These are the people that do the work in the lab. They are all wonderful students, wonderful people, wonderful colleagues. And I don't have time to read all the names, but they know I'm thinking of them and say thank you to all of them and my fundings. And a special thank you to the organizing committee, Dr. Sarah Jeldin, Dr. Rami Aziz, and Mrs. Ezra Raghav and Mrs. Marva. They took care of all of our affairs, and especially you for your attendance, and shukran is the word.
Okay, is there a quick question for Marta as we change over to the next speaker? Yeah, somebody at the back. Yeah.
Greetings, Dr. Marta.
Thank you.
Thanks a lot for your enjoyable presentation. In terms of efficacy of phage therapy on bacteria, you know that there is a great deal of difference between the in vitro experience and in vivo experience. You have discussed the human trials as part of a challenge that you faced, and maybe a challenge in the future. So I want to ask about something. All of us know that certain bacteria are called facultative intercellular. They may be living inside the cell, and they can evade the immune system. Antibiotics, some of them can achieve high concentrations inside the cells. How can this be applied to the case of the phage? Thanks a lot.
Yeah, that's a very good question. All what I work with is related to extracellular pathogens. So I've never worked with intracellular pathogens. But yes, there are some evidence that show that in some way, I really don't understand. You can get phages inside the cells and get into the bacteria that are intracellular. I don't know exactly how it works. I've never worked with that. And the other aspect of your question, yes, we have been working, the mice results I show you, these are lab experiments, yeah? But we work in association with the Shrimp Farmer Association, and they help us to do some work in the field conditions with shrimps. In Colombia, we have a huge problem with the Bacillus licheniformis, which kills the shrimps. And we isolated some phages, and we tested in the aquariums they have, not in the big ponds, but in a smaller setup. And it works really well. So we got some funding for that work, and just recently we got also funding to do some field experiments with the poultry farms. And in that case, you are right, Salmonella can be intracellularly. But in this case, what we want is to control the Salmonella that inhabits the poultry digestive tract. And it's a commensal, so it's living there in the outside of the epithelium. What we want to prevent is Salmonella to get into the meat of the poultry that finally humans are consuming. And this is the problem.
Just time for one more quick question.
Hello, my name is Dina. You said that the phage bacteria is selective to each type of bacteria. Each one has a phage to attack it. So why the cocktail phage have this? They are used to make to decrease the resistance, or they make synergistic effects.
Now, what we think is happening is that the bacteria cannot respond so fast to 3 different aggressions. So she may evolve resistance really quick. I mean, in 48 hours in our experiments, all the population was resistant. And the underlying process probably is what Marta already said. You have like small subsets in the population. One of those is resistant to that particular phage, but probably is not resistant to the other 2 or 3 that you are using in a cocktail. And when you try to use that in phage therapy, you never know what strain is that the one in the patient or the animal that is causing the problem. So it's just a better strategy to use a combination of phages.
Okay, thank you very much, Marta, for a very interesting talk, telling us some different applications of phages. So our next speaker, I'd like to introduce Ayman El-Shibini. He did his PhD in Nottingham University, postdoc in Cardiff and Olympia in the US, and now he has his own position here in Egypt in Zuhail University of Science and Technology.
Thank you, Marcia. First, I would like to thank all the organizers for inviting me to participate in this beautiful and wonderful conference. I'm going to talk and carry on our journey to talk about bacteriophage. And the outlines of my presentation will be mainly about bacterial illness and top pathogens and phage therapy against Campylobacter and the E. coli. And maybe we will talk about other few things which may be interesting for you. We all know about bacterial illness characterizations that there are some different characterizations for pathogens to cause illness. Some microorganisms cause infections. Infection itself is enough to cause the illness, and some other bacteria cause illness by intoxications by producing some toxins. And the third category is toxico-infection, which needs the pathogen to infect the human beings and cause the infection by producing toxins at the same time. And the reason why I mention this is that because I'm going to talk mainly about pathogenic bacteria and how we can control it, mainly the foodborne illness which caused by such pathogens. And as we know that different pathogenic bacteria can cause different symptoms, and this depends on the pathogen itself. So it starts from having fever, diarrhea, and vomiting, and it may cause some problems such as stomach ulcer, or can, in severe circumstances, can cause cancer. And finally, at some time, it can cause death for human beings. The top pathogens, such as Campylobacter, Salmonella, Listeria, E. coli, Bacillus, and Listeria, and I'm not going to talk about all those pathogens, but I need to mention just that most of those pathogenic bacteria are very, very important and are found everywhere. And the reason for their pathogenicity or their dangerous effect on human beings that they can cause serious problem due to after infection, and we can get them easily if we didn't follow the hygienic procedures or the public health steps which we have to deal with such microorganisms with care. So if we look at this figure over here, we can see that those 5 microorganisms, starting with Campylobacter and E. coli, 0157, Listeria, Salmonella, and Vibrio, and we see that most of them are quite stable along this time up to 2012, and by the time we see that Vibrio increased from year to year, and this is actually a big problem for us. Even if they are stable, this is not good enough for our health because they are there and we can be infected at any time. So, how can we get infected with such microorganisms? We can be infected with those microorganisms by handling foods. So there are many different sources. So the food may be infected itself by these microorganisms from different sources like sewage water or any other sources through while we are growing it or while we are processing those microorganisms. And we can get infected by cross-contamination. If we handle or eat a contaminated food, we will get infected with such microorganisms. So this is a big problem for our health. And we all hear that there are many outbreaks with such microorganisms. And I'm going to present only a few examples only for E. coli, which have been reported in USA. And those, in 1993, there was an outbreak. People eat sandwiches from Jack in the Box restaurant in Washington State, and about 732 people get infected and get ill, and only 195 go to the hospital and hospitalized, and 4 people died from this infection. And if we look at the other examples, you will see that every time we have some people die from such infection. The latest outbreak was in Germany, as we all hear about it 2 years ago, I believe, in 2011, and it was with a new strain. And this new strain, which is E. coli 0104, H4, and it had a bad impact on the population because more than or almost 4,000 people got ill with this infection, and 53 of them died. So, the problem, as Marcia mentioned, the only option we had at this time, and even now, to use antibiotics to treat such pathogenic bacteria. But as Marcia mentioned earlier, that those microorganisms can acquire resistance very, very easily. And this resistance due to the mutation in DNA of those microorganisms. And once we get one colony mutant or resistant for such antibiotic, it has the possibility to grow and grow and grow. And you know, in 8 hours we can get up to 500 million bacteria if they have the right condition to grow very well. What is the problem with such antibiotic resistant bacteria? It can spread everywhere. So we can have it from animals, and we can transfer it to animals, and we can transfer it everywhere around our environment, which is a serious problem for ourselves. So if we get infected with such multi-drug resistant bacteria, this is a horrible, it will be a nightmare for all of us. And to develop a new antibiotics, it is not easy. It takes years to handle such problem. So, what is the solution? The solution from our point of view is to use, at certain cases, phage therapy, especially with food. And the applications of bacteriophage as a phage therapy, it has been used to, in food safety, to improve the food safety, and also to enhance the animal production and the animal growth. And also we use it in cleaning and disinfection of any service, especially in hospitals, and also in human therapy. So, I will mainly talk about the animal trials, which we use, and especially with on Campylobacter. So, we always, as Marcia mentioned, that we isolate phages from everywhere. It is everywhere in our environment, and it lives even inside our stomach. So we can isolate it, and the most important source for isolating bacteriophage is sewage water, because it is there with its host. So each bacteria has its own bacteriophage. So to isolate your bacteriophage, look in the same place where is your bacteria live. So with Campylobacter, we isolate Campylobacter bacteriophage from poultry, mainly from chicken, and it lives everywhere in poultry, Campylobacter. And we also isolate bacteriophage from the same source. Then we amplify the bacteriophage in its specific host, and then later on we do the in vivo experiment to see how this phage works. After we isolate the phages, we do the lytic pattern to study how many bacteria are sensitive to those bacteriophage. And this is the bacteriophage. I draw this phage on the surface of E. coli, and it is a T4 phage. And the reason why I present this bacteriophage, if you look at those bacteria grow in the clear zone. This is the resistant one, which Marcia mentioned a few minutes ago. After we isolate and study the lytic pattern of those isolated bacteriophage, we do some electron microscope images to see how the structures of those bacteriophage and which families they are related to. And later on, we did some experiments. I'm going to present one of the most important results we got when I was there in UK. So to reduce 2 log of pathogenic bacteria is a great effect to reduce the risk of infection with such pathogenic bacteria. In this paper, we actually succeed to reduce the risk or the log by more than 5 log. 5 log reduction with Campylobacter jejuni numbers. And as you see, this is the control, the white circle, and this is the black circle, the one are the treated ones. So this is a drop of the numbers of Campylobacter after phage treatment. Due to the time, I'm not going to go in more details about all the experiments we did. So this is another bacteriophage, and it is, we call it can be the group 1, which is a larger size of bacteriophage. It is about 190 kilobase per, and it's quite large. And we did the infection for both Campylobacter jejuni and Campylobacter coli. They are 2 different strains. So with Campylobacter jejuni, we could achieve 2 log reduction, while with Campylobacter coli, we couldn't achieve much reduction with this bacteriophage because Campylobacter coli behave differently from Campylobacter jejuni. So other people have studied the MOI, which is the multiplicity of infection. How much phage do we need to use to control or to kill such pathogenic bacteria? Do we need to increase the number of bacteriophage or use low or use low number of bacteriophage? So people did a lot of experiments to explore such important factor, and they found different results. And the reason for this, sometimes, as Marcia mentioned, that it is important to use phage, a cocktail of phage to compete or to get rid of most of bacteria. And the reason for what is for this is that you can use different bacteriophage with different receptors. So they can recognize the bacteria with different receptors, so to attack it and kill it very quickly before it develop the resistance. القطرة تغلب الشجاعة يعني بالعربي. This is an experiment that people used 1 phage and used two phages, and they couldn't see major differences between using a cocktail and the single phage. And the reason for what? That sometimes those different bacteriophage in a cocktail interfere with each other, so they can affect each other. But this is not common, but it can happen. This is what Marcia mentioned, that some bacteria can develop resistance by the time, and this is an important issue when you deal with phage therapy. This is a recent publication which we did at Evergreen State College. So we used 2 different bacteriophages to infect E. coli, and we got great results because those 2 bacteriophages can kill most of the bacteria, and we at certain time we can't detect any more bacteria. And other people could reduce the number of E. coli to undetected limit. So it goes to the undetected limit, and they couldn't isolate any E. coli strains from their treatment. And this was done in milk. So the question is it good to use? Yes, in some certain cases, yes, we can use it to kill most of the bacteria, like we see in this results. So, in a brief, as I mentioned, MOI is different. One phage or one isolated bacteriophage can kill most of the bacteria at low MOI. We need only few numbers. They are strong enough to kill this bacteria. And some other bacteria, we need more or many high MOI to infect such microorganisms to achieve higher reduction. This is a nice work done in 1982. They used different antibiotics to treat E. coli in mice, infected mice with E. coli 018. And they found that bacteriophage, when they treated the mice with bacteriophage, phage R, only 2 mice have died. And this is only with 1 dose. While with antibiotics, different antibiotics, most of mice died due to even after the treatment with antibiotics, which is quite promising and amazing. So I'm going to present some other nice picture. What is the future for using bacteriophage? Is it only for animals trials or for food, or we can use it for human being? In Tbilisi, they use it to treat human infections, and they have many different products like this one. And it is in the market. They produce the phage in vials and they sell it. And this is one case that they treated someone. I know that it is not a nice picture. And this is a recent one which they treated this diabetes with bacteriophage and they recovered in a good time. This is the most common companies. FDA approved the use of bacteriophage to treat animal infection. UK approved the use of bacteriophage to treat listeria. Even they spray the fresh vegetables and fruits. And there are many companies. It is a promising approach to adapt. And finally, I'd like to thank you for your attendance. Thank you.
Thank you very much, Ayman. Is there a question or two that we could take for Ayman? Yep, at the back.
Assalamu alaikum. First of all, thank you all the presenters in this session. My question is about Egypt, and you are Egyptian. Is there any products, or are there any products of these that enter the Egyptian market? And if yes or no, what are the obstacles or threats that can oppose such a spread of this new approach? Thank you, Doctor.
Thank you. Actually, we are only few people in this area in Egypt. We are about 3 or 4, and all of us are here, actually. I wish there are many others, but we are only 4 right now, that's what I know. And there is no obstacles, and we have started to collaborate with each other very soon. Inshallah, and I started my own project at Zuhail City of Science and Technology, and I hope that we are going to produce a product soon, inshallah, to help the diabetes to. I can't give more details about this anyway. Sorry. So there is no any obstacle to deal with such phage therapy approach. Any other questions? Okay, thank you very much.
Would you please? I have a question.
I can't see.
I'm here.
Sorry.
In fact, I have 2 comments and questions.
Yes.
In 1982, I could produce cancer of the bladder in hamster using Escherichia coli isolated from cancer bladder patients carrying Bacteriophage T7 and another lambda. And that was in the United States. And in hamster was the same percentage being in Egyptian patients, which was 28% in hamsters. In fact, this is published, and I got the membership of American Society of Microbiology. The point is what I am annoyed by using the bacteriophages for therapy because these are genomes, and genomes is not, it is dynamic, is not static, and it can produce another mutations in the cells treated, especially in viable people. For instance, like treating of chicken or something like that, it's okay, but if you inject, you can treat something, but you introduce some genetic materials you don't know. Also, what I didn't like, that you didn't nominate the type of the bacteriophages you used and what is their lytic or lysogenic. And that is not scientific, in fact. You showed the electron microscopic picture without the type. The other thing, I think that you need to make sequencing. And in England, for instance, they used the bacteriophage therapy for including genes which can replace some deficit in some genetic material produces diseases. And that was long.
I'm sorry, we haven't got
Habib, who was the first to do that. So I thank you, and also for the lady whom also she presented. It was a good talk, but it needs to identify especially the type of the viruses used, because this is our viruses. Thank you.
I think in the interest of time, perhaps you could meet with us after and we can answer some of you had a whole list of interesting questions there. I think we ought to move on, however, to the. Do you want a quick response, Eamon, or should I think?
I can answer all the question later on, but again, I just want to answer the second one. That so far there is no any side effects for phage.
Since in 80s,
I think we haven't got time for this discussion. I'm very sorry. We have to move on.
In Clostridium Hospital, and the work on bacteriophage, and in Ain Shams University, and there are not 3 people in the whole Egypt and are existing here. Thank you.
Thank you. We can. I can answer your question later on, because of the time. Yeah.
No, I'm sorry. We don't want to overrun the session. So thank you. So our final speaker of this session is Hani Anani, and he's been working at the University of Guelph for a number of years now, and he's been doing some very innovative work on working out how to actually get bacteriophages to where you need to deliver them to in a therapeutic setting. So he's going to tell us about that now.
Thanks, Mos. So Assalamu alaikum first. So as I'm the last one, so can stretch a bit. And I hope that my tone of voice actually will wake you up, all of you. So first of all, I just would like to thank the organizers actually for inviting me to this interesting meeting and give me a bit of time away from the cold weather in Canada. So enjoying such a beautiful weather in our beloved country, home country. So what I'm gonna go today through is about different method of application of the phage. As you have heard actually from the previous speakers that it's a promising tool and it's been here for a long time. So our objective actually is just to broaden the application of the phages in different areas. So just as Dr. Ayman mentioned about the foodborne illnesses, so I just would like to give you a bigger image about numbers that for the whole foodborne pathogens or the foodborne diseases. And as you can see here from the numbers here that you have millions of cases every year are reported in the United States and Canada, and this costs as a consequence millions, billions and trillions of dollars every year for both countries. So it's not only a problem in the developing country, but it also in a developed country. And when you go to the recent epidemiological studies showing that the major organisms that are causing the foodborne illnesses are viruses, and bacteria actually come to the second to the viruses. So bacteria represent a major etiological agent that can cause the foodborne illnesses. So everybody wants to fight bacteria, and through two ways, we can just biocontrol or inhibit the growth of the bacteria on the food samples, on the animal-producing animals, or detection. So we can avoid the recall, the big recalls of the food products, and save money for the company. So here I'm introducing a new term in this session, which is a detection, using the phage as a detection. So I'll speak about using the bacteriophage as a biocontrol tool to inhibit the growth of different pathogens, and also to detect our approaches that we have developed in Guelph, University of Guelph, Canada, about using the phage to detect various pathogens. So as it was mentioned before, we have two cycles, lytic cycle and lysogenic cycle. When you are dealing with the biocontrol, it's better to use the lytic phage for obvious reason, to avoid the development of more virulent genes actually when if the virus is lysogenic phage. So all the viruses that I'm going to speak about that were lytic, some of them, most of them actually were sequenced to ensure that they don't have the integrate gene or any lysogenic related genes. So our lab actually is part of a network. It's called Sentinel Bioactive Paper Network, including different universities and industrial partners in Canada. And the major goal of this network is to develop bioactive paper or bioactive solid material that can be used for the biocontrol or detection. And as we are working with phage, our aim is to develop bioactive material, packaging material that can be useful actually for wrapping the food, enhancing the safety of the food, avoiding the perception of the consumers about eating the phage. Because currently, as it was mentioned earlier today, FDA has approved the application of Listeria phages on the ready-to-eat meat, but the way that they are applying it right now is by spraying. So in order to just gain some of the consumers' support for this application, we aim to develop bioactive material that can be just used for the packaging, and before eating you can just remove the package, and supposing if there is no bacteria there, there will not be viruses there. The other application is about the detection and developing the handheld device. Okay, in the poor countries or even in the developed country, you need fast result, not with a highly sophisticated technique actually to detect the bacteria as fast as possible. Also, this will broaden the application of the virus to other applications, and we are currently thinking about broadening that to, for the bandage, for example, for the wound dressing, for the water filters. So once we can achieve the immobilization on any solid membrane, we can apply it and achieve the stability of the phage for a longer time. So when you think about immobilizing the phage, actually, it comes that the orientation, as you got from the previous lecture, it's very important to have the virus have the tail fibers free to be able to capture the bacteria. Otherwise, the infection will not occur. So there are some receptors that should be bound to the tail fiber protein here it comes the specificity of the bacteriophage to start the infection. So we started a long time ago to do some genetic modification for one of the well-known viruses, the T4 virus, by the phage display, by inserting some proteins, actually cellulose binding domain to the head of the phage, and here is this toy actually. So we put the protein, this is my beloved toy. So you can put the cellulose binding domain here, so we can direct the phage to bind to the cellulose membrane or any substrate specifically. So it will be oriented to the support of the support that you are using. But however, this actually is took from the student who did it actually about 5 years just to do that, and unfortunately it affected on the percent size or the infectivity of the virus. So we started to think about more broader, more general approach that we can use for different bacteria or different pathogens, different phages, sorry. So we started to think about the physiosorption, but with the oriented immobilization as well. So we asked ourselves, is there a charge difference between the head and the tail fibers? And we know that the overall charge of the bacteria is negative charge. So could be the charge is playing a role actually of attacking the virus to the surface of the bacteria. We got a mutant of T4 viruses that can produce head components only and tail components only. And we measured the zeta potential, and we found that there is a significant difference actually between the head components and the tail components. So we asked our collaborators to design for us positively charged membrane or beads, okay, and we immobilized the phage on them to have the tail fiber free so it can capture the bacteria and cause the infection. So in our lab, we are starting by the characterization and isolation of the phages for sure. It's the first step. We need our raw material. And then we are going to the immobilization step. And then using the immobilized phage as a biocontrol tool and as a detection tool, as I will explain it later actually in my talk. So the phage isolation and the characterization. You heard enough about the phage isolation and the characterization and how it's important to pick it very good. So I usually and briefly summarize that in the coming slide, just also because my kids love it. So it's super phage. You have to look for the super phage, okay, that can cover all the criteria actually that was mentioned in the previous lecture. There is, try your best, I'm telling my students, try your best to get this super phage, host range specific, and know your downstream application. If you are going for the biocontrol, you have to pick the broader host range virus. If you are going to the detection approach, you need to be very specific and make sure that it's targeting your target only. So we are working with different, so many types of bacteria, and I would say that right now we have a fair amount of virus against different bacteria. Most of them are characterized and sequenced, and we can start the second part, which is the immobilization for the biocontrol application. We tested different bacteria. I'm not going to go to the details because of for the sake of the time, and I would like also to focus on the detection part. We tested them against the Listeria monocytogenes against ready-to-eat meat and cantaloupe, E. coli 157 against raw beef, and E. coli 104, which, as Ayman mentioned, the one that caused the Germany outbreak. We got this strain, we isolated the phage against it. We encapsulated the phage in this one, and we applied it to the alfalfa seeds and the sprout, and we got good results, and I can show them to you later. The challenge actually for the food application, as if we come back to the first question that was asked here in this session, that in vivo application. So in vivo, when you speak about the human, yeah, it's in vivo. It's, the result actually could be sometimes not very good. But here also there's a big challenge of application the food in the food matrix. Very, very challenging experiment. However, I will just summarize briefly one of the experiments to show you what we are doing. So we have the cocktail of virus, and it was mentioned before that using a cocktail of virus is better to reduce the chance of the developing of the resistance. And we immobilized them on the positively charged membranes, and then we added them to a previously inoculated raw beef samples. And we incubated that at 4 degrees, and we found that there was a reduction in the count of the bacteria in the treated samples until we reached day 12, and the count was below the detection limit. So it was successfully applied and working actually in the immobilized form. When you use a strain that can produce luminescence, we found that there is, when we apply the phage, you cannot see any luminescence appearing actually on the surface of the meat when it's incubated with the membrane or the immobilized phage, while you can see that luminescent bacteria can grow and produce luminescence in the absence of the phage. So it's working, and it's a promising technique, but again, the challenge of the food matrix and just to find one bacteria over the surface of the food sample is a lot of challenges actually that you need to think about it, in addition to the regulatory application, regulations as well. So the second part of my talk actually is about the using of the phage as a detection tool. Detection actually in the food, it's again very, very challenging because you are just looking in a haystack, of a needle in a haystack. So just normally if you are following the good manufacturing practicing in producing and processing your food, you end up by having 1 to 10 cells, okay? And there are some bacteria, there are some regulation, or the regulation in USA and in Canada, 0 tolerance for them. Like for example, Listeria monocytogenes. You shouldn't be able to find single cell in a ready-to-eat meat. So that's very challenging. How can you get that? So the traditional method of doing that is to increase those needles. by doing enrichment for another day, incubating them another day or 1 or 2 days, so the bacteria will increase in number, so you can cultivate them and use the traditional method or some molecular technique method actually to identify them. Here it comes the role of the phage that can be a promising actually to enhance and do the amplification. One phage can infect the single cell of bacteria and produce about 150 to 200 particles. You're increasing your signal. So if you detect the phage, you're increasing your signal by 2 orders of magnitude. So this is the assay that we are looking for. Very challenging. You need 4S assay, which could be the name of the company that I could open later on after I'm done with this academy. So it should be simple. So we need simple handheld device that anyone can use. Short time, can be done within a working day because the company needs to deliver the product to the market actually as soon as possible without holding it. So we need to track that. And specific and sensitive. And the specificity here is a fitting criteria with the phage. So just let's do it. I will explain to you what we are doing exactly. So first of all, the phage, when the phage mixed with the bacteria, the infection will happen, or the absorption first, and then the infection. And as a result, all the cell will lyse. As I told you, we are working with a lytic cell, so within 20 minutes or so, for most viruses, you can have the cell lysed, and progeny phages actually can be released. So you have 2 things here. The progeny phages released from the cell, which could be a large number, so you are starting by just 1 cell. And as I told you, you end up by having 200 particles released at average. And some virus can produce more, up to 1,000 particles actually. And the other thing that could be released from the bacterial cell is enzymes, which also large number of copies inside the cell, not only 1 copy. So you are doing amplification here. You are increasing the number of the cell. If you are looking for a single cell, you are increasing the number of your target right now to be about 200 or more, actually hundreds of enzyme particles that you can detect. So when you look to the progeny phages or the produced phages, okay, you can detect them, you can sequence, you determine a certain unique sequence, okay, and then you can apply any molecular technique such as polymerase chain reaction. And then you can apply it, and also you can design an ELISA-based technique, which we are doing right now. You can develop a certain specific antibody against certain protein on the phage specific, so we can develop ELISA-based technique colored so we can detect the progeny phage. The other part actually is just to add the indicator, substrate, and then colored product. So we'll start with this part. So I hope that you got the point that we are increasing the number, or we are amplifying the signal here. So we'll start with that, and this is we use for detection of E. coli, fecal E. coli, to determine the fecal contamination of the water actually. You know, all of you know that it's coliform bacteria, or especially E. coli, can be used for the detection of the fecal contamination of water. So we filtered the sample. We put it in this device. It's a small handheld device we developed in collaboration with University of Alberta. It's made of special material. It's a handheld device. We got the filter and we put it here, and then we incubate it for just 4 hours to increase the number of the bacteria. And then we add the substrate to detect the enzyme and detect the color. We can add another substrate actually that can, instead of producing color, produce luminescence. And you can do that. And here's the result. We reached up to single cell detection limit with a colorimetric approach and with the luminescence approach. And currently, we are designing a mobile app that actually you can have this device, you filter your sample, and you can just measure the color, measure the intensity with the mobile app, and can give you a semi-quantitative reading while you are in your place. And within, as you can see, 8 hours or 5.5 hours only. The other part is just by the real-time PCR, using the real-time PCR. So again, mixing the phage with the paramagnetic beads, and should be immobilized actually through their heads, as we explained before, and mixing them with the bacteria, allowing them to infect, absorb first, and giving them some time, up to 5 hours to have the infection. And then with the magnet, you can remove the originally added phage, so you can enhance, you can just detect the progeny phages only with the real-time PCR. And with that, actually, we were able to detect up to from 5 to 10 cells, E. coli 0157 within 8 hours. And we are currently trying to optimize also this approach. But we need to go to the handheld device, something that we can provide it like the pregnancy test or any test, add your sample, see the color. So we moved to, instead of using the expensive machine of real-time PCR with $30,000 actually to get it, so to another technique of amplification similar to the real-time PCR, exactly, but amplifying the DNA at one temperature. So there is no need for fluctuation in the temperature, 65 degrees, and then it's high efficient, high specific, and efficient, okay, and doesn't require special equipment. We started, and we can detect actually the product of this lamp or the isothermal DNA amplification through the luminescence light or by adding a certain reagent to develop the color. We started with some viruses, and we were able to detect 10 to the 3 BFU or black forming units of the virus against Salmonella and the E. coli 0157 within 60 minutes. And currently, we are doing the bacterial experiment. So last thing that I would like to mention to you about toward large scale production. Okay, I can do that in the lab right now, but we need to approach the industry. We need to tell them we have a method that you can use it. So we started to coat the virus using coating machine, normal coating machine for the virus. But unfortunately, due to the high temperature, this killed the bacteria. But recently, actually, we were able to use just the inkjet printer that you are using in your office, but a different design that doesn't have heat in it. And we were able to print the phage on a paper. So, and it was able, when we mixed it with the host cell, we were able to see a good reduction actually when we mixed it with the host for E. coli, Salmonella, and Listeria monocytogenes. So in conclusion, I would say that, and hopefully after the all talks that you have heard today actually, that phage is a promising tool for not only therapeutic as a therapeutic tool, but also as a detection tool. And having the phage in the immobilized form or encapsulated form will be more economic application for the virus, will broaden the application of the virus. So in my opinion, as a phage lover, that the next generation or the next decade actually technology would be the phage, not only as a biocontrol, but as a detection tool. And finally, I would like to thank our collaborators from different labs actually across Canada, and our funding agencies, the Sentinel Bioactive Paper Network. And I would like to thank the phage team in our lab, Dr. Mansell Griffiths and Dr. Luba Brovko, and all the hidden soldiers actually behind all these projects going on in Griffiths. Thank you for them all, and thank you all, and I'll be happy to get you.
Thank you very much to Hani and to the other speakers for very interesting and diverse set of talks. Are there quick questions for Hani? While you're thinking of your questions, I think Ayman wanted to have a chance to answer.
Yeah, if you have any question to any of us, so we can.
A little bit, the last question, and then we can take a couple of general questions if anyone has any.
Yeah, regarding what kind of phage we use, we never use lysogenic phage for phage therapy. We always use lytic phage, so which is quite different from the lysogenic. Lysogenic is not acceptable to be used in phage therapy. And I know that sometimes some lytic phages at certain circumstances can transfer to lysogenic and vice versa. The other thing we always, I didn't mention the name of the phage because it is a general talk, but it is a Maveridish family, and it is a lytic phage which we use to treat such pathogenic bacteria. Regarding the immune system, the phage itself is present in our stomach. So it is there because our stomach has microbiota, which is different bacteria. And once you have the bacteria, you have the bacteriophage. That's why we isolate it from the sewage water. And so far, there are some recent publications that they reported that phage can support the immune system for some extent. So there is no any side effects have been reported since they discovered bacteriophage until now, and that's why people in Tbilisi inject it to treat such pathogenic bacteria, and they didn't find any side effects until now in Poland and in Tbilisi as well. I can't remember the other question, but yeah,
Okay.
that's.
Are there any other questions for all of the speakers? Well, we're very approachable, so if you have some questions you'd like to ask us later today or tomorrow, please feel free to come and talk to us. Otherwise, thank you very much for your attention.